Hi, my name is Aaron-Emiliano, and I am a biotechnology major at Holyoke Community College, as well as an NSF STEM scholar. I am excited for this opportunity to present my research project at the Umass Undergraduate Research Conference, and I can't wait to see the projects completed by other students as well. The title of my project is Sickle Cell Disease through 3D Representation, a model for adaptive learning environments. Standard pedagogical methods do not account for the barriers commonly faced in academic settings by those who have difficulty with standard learning styles such as exclusively verbal or literary instruction. A complex topic such as gene expression and the effect of mutations on disease state can be difficult to both teach and learn. As a person who is affected by learning disabilities, I chose this project in order to help myself better understand the subject, as well as to improve the accessibility of education to others with similar needs. To achieve this, I'm developing a comprehensive lesson plan in relation to sickle cell disease. This is framed around the topic of mutations and the implications of genetic mutations on protein structure. I am also incorporating a physical component for better visualization. This physical component consists of 3D models of both healthy and diseased hemoglobin proteins. These models demonstrate the connection of multiple topics including genetics, mutations, protein structure and function, and disease states through the use of tactile models and real-life examples. In the future, I hope to also include an example case study and CRISPR component. So what is sickle cell disease? Sickle cell disease is an autosomal dominant genetic mutation that occurs within the hemoglobin protein. This disease causes red blood cells to deform into a sickle shape, which is known to cause chronic pain in people, known as vaso-occlusive crisis, as well as organ damage or failure. Sickle cell disease is caused by a point mutation that replaces glutamic acid in an amino acid chain with valine in its place. The hydrophilic and hydrophobic specifications in the picture refer to the change in the structural shape of the hemoglobin. The glutamic acid on the beta chain in the normal hemoglobin is hydrophilic. In the case of hemoglobin S, when this glutamic acid is replaced by a valine, it is then highly hydrophobic. Since the beta 6 position is located on the surface of the protein, its shape is drastically affected by its surroundings. The normal hemoglobin is able to maintain the proper structure as it bonds with the water around it, and it remains stable. Since hemoglobin S is so hydrophobic, it ends up binding to another hydrophobic hemoglobin. This hydrophobic reaction is what causes the hemoglobin clumping that leads to the sickle-shaped blood cells. The video attached from the RCBS protein databank displays the valine 6 point mutation in the 2HBS, also known as hemoglobin S. Hemoglobin S stands for hemoglobin sickled, which displays the sickle cell disease mutation. Protein databanks can be a little difficult to navigating and can be intimidating at first. I believe that using 3D models as a visualization aid would make it a bit easier for people to understand these molecular concepts portrayed on the protein databanks. On the left, you will see a model representation of a mutated hemoglobin protein, hemoglobin S, as we saw in the video above. This handheld model can provide a very clear visual of what sickle cell disease represents like in a hemoglobin. Here we can see the elongated fibers caused by the hydrophobic reaction that occurs in hemoglobin S, or 2HBS. To the left, you will see a video of hemoglobin A, which is the most common form of hemoglobin found in the average adult human. This model is defined as 4-HHB in the RCBS protein databank. As done with hemoglobin S, I hope to also 3D print a model of hemoglobin A in order to have these models be able to be held side by side for visual comparison. My goal for expanding upon this project is to also produce a 3D model representation of the primary structure which becomes mutated and creates the change in the final protein structure. This model represents a clear example of the point missense mutation in the primary structure of the protein. The codon sequence is altered, and this alters the amino acid sequence. This is where the glutamic acid is changed to a valine. The alteration of the primary structure of the protein leads to the mutation of the quaternary structure, the final hemoglobin protein, which is 2HBS. I believe that 3D modeling can be used in a wide variety of classes to better display concepts being taught. As an extension to the primary and quaternary structures we discussed above for these proteins, we can also display secondary, tertiary, or globular and fibrous protein structures. We could also create molecular models to display structures and interactions occurring in a chemical reaction. 3D printing could be used to create anatomically accurate organs for biological instruction, such as a heart, kidney, or brain. Fungi and plant models can be made to accurately depict differences in structure, size, or texture. Models could also be created to upscale microbes and cells to demonstrate structural components. 3D printing could also be used to create models of extinct, endangered, or otherwise difficult to acquire specimen for study. Throughout the process of 3D printing these proteins, I did face some error. I had many failed prints before I was able to have the one successful one seen in the picture above. I hope to continue with the 3D printing process and in the future produce both successful prints of the normal hemoglobin and the sickled hemoglobin. For this project, I used a variety of softwares, 3D printers, and materials to learn how to best 3D print the proteins. Some of the softwares I used include Fusion 360, Ultimaker Cura, FlashPrint 5, Mesh Mixer, Chimera X, Avogadro, VMD, and MeshLab. I also used a variety of 3D printers to test which one was best able to print these complicated molecular models. I used the Ultimaker 3 Extended, the Creality CR10S. The materials utilized included PLA filament, PVA filament, and 3D Fuels HydroSupport V2 water-soluble support filament. I'd like to thank my mentor, Professor Emily Rybinski, for her continued support, direction, and for helping me in developing my confidence. Professors Ileana Vasu, Laurel Carpenter, and Vanessa Martinez for all they do for the NSF STEM scholars. Melissa Patchouli for making all my 3D printing dreams possible. Camille Close for her endless support, reference aid, and always being there when I need her. And the ever helpful people at the Umass All-Campus Makerspace for answering my technical questions. Thank you for your time and listening to my presentation.